My studies have brought me near to a host of biological horrors that many have never dreamed of, much less thought to be real. Perhaps the most notable of these studies have been pathogens: the human vampiric virus, lycanthropic parvovirus, human zombism virus, and others. Each of these contagions resculpts its victim into a grotesque parody of the human form, its medium not of clay, but of bones, sinew, and flesh. Indeed, each virus is a reminder of how delicate our humanity truly is. But individual studies of these viruses, though important, could only ever form an incomplete picture. I knew that to truly understand them, I needed to observe and document their interactions with each other. Now, my valued listener, I will present to you the outcome of an extended study into just what occurs when a victim of lycanthropy is exposed to the virus that leads to zombism. But I will warn you, the results may not be what you expect. Following my observations of specimens singularly afflicted with lycanthropy, and of those with zombism, there was still much to be discovered. I do not have the resources I once did, but I knew that if ever these diseases were to become widespread, the possibility of a co-infection with one or more of these cryptic pathogens could lead to even more catastrophic effects than either could achieve on their own. And so I set out to obtain specimens for my research. This was more difficult than it sounds, as my studies of lichens in particular had previously only been the result of deceased individuals. But even now, I do still have some connections. And after some time, a few willing participants were procured. Now, before I proceed, I should clarify that in most cases, the mental state of those individuals affected by LPV quickly deteriorates into something animalistic. There is no cure, nor can there likely be one given the complete remodeling not only of morphological structures, but of cerebral structures as well as a result of the viral infection. Participants in this study were fully informed of their situation, having been exposed to the LPV in some form prior to their recruitment, and were given the choice to proceed. Their contributions to this critical research will be forever remembered. I have spoken at length regarding LPV and HZV in previous recordings. However, a brief survey of these viruses is warranted for this purpose. The lycanthropic parvovirus, or LPV, is a novel strain of zoonotic virus belonging to the family Parvoviridae, having specialized to infect both canines and primates. Like others of its family, LPV is a non-enveloped single-stranded DNA virus, but in contrast to its relatives, LPV does not destroy host cells. Upon entering a cell, the viral DNA immediately integrates itself into the host's chromosomes, and new LPV virions bud from the host cell, leaving it intact though altered. Within a week post-infection, the victim has begun to exhibit the hallmarks of the lycanthropic appearance: widespread hypertrichosis or hair growth, violent behavior, degradation of communication, and the early stages of an agonizing reshaping of bones and soft tissue, the latter of which many do not survive. Those that do are entirely transformed, for lack of a better word, within about 6 weeks. The end result is a human stripped of their higher brain functions, primarily due to a reduction of the prefrontal cortex and hippocampus, with a pronounced mesocephalic snout and a primarily quadrupedal form of locomotion. Notably, and contrary to popular notions, these changes are not tied to any lunar cycle, nor can they be reversed. The human zombism virus, on the other hand, works very, very differently. It belongs to mononegavirales, the same family as those that cause rabies and Ebola, as well as vampirism. Like LPV, it is zoonotic and often transmitted via bites. Unlike LPV, however, it is a negative-stranded RNA virus and utilizes a reverse transcriptase enzyme to produce DNA from its own RNA genome. This new DNA is then absorbed into the host's nucleus and incorporated into its genome via an integrase enzyme. This effectively makes it a provirus, transforming the cell and directing it to synthesize new virions. Of course, HZV's replication strategy is inherently destructive to the host cells, leading to widespread cellular damage, necrosis, and the near-complete breakdown of bodily functions. This is followed by a rapid decline in the host's physiological conditions, with symptoms of aggression, decay, and cognitive decline evident within mere hours. Victims of HZV enter a comatose state approximately 1 to 2 hours following exposure to the virus. In this state, blood flow, respiration, metabolism, and even cardiac function are reduced to nearly nothing. Brain function is also greatly affected, and in fact, the victim frequently appears deceased. Following this phase of infection, the victim reawakens, though the heart has ceased to function entirely. Instead, skeletal muscle performs the role of blood circulation, albeit poorly. As a result, widespread organ failure and tissue necrosis is rapidly evident. Ultimately, these two viruses employ strategies that could almost be considered opposite one another, and this is precisely what makes this case study of particular interest. In the natural world, the concept of co-infection is not only common, at times it is thought to be more common than single infection. In these cases, the two agents of infection often, paradoxically, compete with one another while simultaneously increasing both agents' rate of infection by overwhelming the immune system. In some cases, the presence of two pathogens can be a net positive for both, especially in terms of transmission and progression. This is a concept known as syndemism. But the interactions between HZV and LPV cannot be so neatly categorized. At the cellular and biochemical level, these two viruses engage in a fierce competition for the host's cellular machinery and resources, often negatively impacting their respective pathogenic processes. LPV, aiming to integrate and express its genetic material within the host's DNA, relies on the host's transcription and translation systems for the production of viral proteins necessary for the lichen transformation. This process demands a significant share of the host's nucleotides and energy resources such as ATP, as well as the use for ribosomes for protein synthesis. HZV, on the other hand, replicates its RNA genome by commandeering the host cell's ribosomal machinery to synthesize its viral proteins, a process that similarly exhausts cellular ATP and nucleotide pools. The primary clash appears to arise when both viruses simultaneously attempt to utilize these cellular resources. Eventually, the host cell simply cannot adequately support the replication and protein synthesis demands of both viruses. As I have observed firsthand, this competition can result in impaired replication for both viruses, disrupting the typical progressions of transformation and resulting in grotesquely mixed phenotypic traits. Put simply, this particular co-infection is nothing short of a biological horror. Given my current lack of resources, locating a suitable environment for study proved nearly as difficult as procuring specimens. But with the aid of my assistant, we eventually stumbled upon a decrepit, though large and isolated, warehouse. Observations were conducted over a period of 6 months. Sample size included individuals of roughly the same age, late 20s to mid-30s, and generally considered to have previously been in good health. This study followed 3 phases, each based on the level of LPV infection in the victim and the progression of the associated symptoms. Phase 1, as one might expect, introduced the zombie virus to a host in the very early stages of LPV infection, within 2 to 3 hours following an infected canine's bite. At this point, the LPV has begun to integrate its genetic material into the host DNA, but the spread of the virus has not yet reached every tissue, and the genetic changes that will result in the lichen phenotype have not yet been expressed. At this stage, the host is usually experiencing generic symptoms, such as fever, chills, increased metabolism, etc. The introduction of HZV at this stage disrupts LPV's entire infection process. Due to the zombic virus's ability to aggressively hijack the host's cellular machinery for protein synthesis, it effectively monopolizes the resources necessary for both its replication and the expression of LPV-induced alterations. In short, at earlier stages, the human zombic virus simply outcompetes the lycanthropic parvovirus, halting the eventual lupine transformation. Instead, the death-like coma indicative of an HCV infection will ensue, though generally delayed by several hours, and the host will likely awaken as usual. Interestingly, however, the semi-intelligence that normally lingers for a day or so in a traditional zombie infection is absent when the host has previously been exposed to LPV. Instead, the victim will awaken with immediate and extreme aggression and exhibit increased locomotive speed when compared to standard HCV victims at this stage. Of course, the combination of the arrested heart and gradual systemic necrosis will take its toll as usual, and so within approximately a week, the co-infected morphology is indistinguishable from that of single HCV infection. In the later stages of LPV progression, usually after roughly 6 weeks post-infection, the lichen transformation is largely complete, resulting in the hallmark canine-like appearance, hypertrophic musculature, and the loss of higher mental faculties. Phase 2, then, was the introduction of HCV in victims averaging 5.5 weeks following an infected LPV bite. During this transformative phase, the increased production of growth hormones, epidermal growth factor, fibroblast growth factor, and insulin-like growth factors promote rapid cell division, muscle and bone growth, and enhanced tissue regeneration. Provided the lichen is able to feed consistently, this elevated hormonal cocktail circulates throughout the lichen's body indefinitely. As a result, if the human zombic virus is introduced at this stage, the cellular damage it induces in its host is naturally counteracted. In my findings, this occurs in several ways. First, through its integration into the host's genome, LPV appears to upregulate genes involved in the DNA damage response, which promotes the repair of the multiple forms of genetic damage induced by the zombic virus. We see a clear example of this in the victim's neural tissues. You see, generally speaking, HCV exhibits cytopathic effects on these tissues, leading to neuronal cell death. It also affects these tissues indirectly, inducing a chronic inflammatory response, the latter of which contributes to further neural damage and dysfunction. HCV's replication within neurons also disrupts normal cellular functions, leading to apoptosis. The inflammatory response exacerbates this damage, potentially leading to demyelination and the loss of neural connectivity. However, notably, the previously mentioned elevated levels of growth factors in the LPV host actually stimulate neurogenesis and the repair of both neurons and glial cells, in a limited way counteracting the mental degradation caused by HCV. Similar enhanced repair mechanisms can be observed in multiple tissue types, which serves to slow the necrotic damage induced by HCV at the systemic level. Notably, no cardiac arrest occurs in victims at this stage, nor does any kind of coma, a hallmark of traditional HCV infection. However, it should be noted that while the effects of the lichen virus do work to counteract many symptoms normally brought on by the zombic virus, HCV infection is not so easily defeated. The success of a lichen fending off the HCV appears to depend on multiple factors, beginning with viral load. In short, it appears that an exceptionally high concentration of HCV can overwhelm the host's cellular machinery and immune response, outpacing the regenerative processes induced by LPV altogether. And though additional long-term studies will be necessary to confirm, it seems likely that over time, the continuous battle between regeneration and degeneration will deplete the host's resources, allowing HCV to gain the upper hand, so to speak. This possibility isn't unique to this phase, however, and so we will return to this concept momentarily. Physically, late-stage victims of this particular co-infection also exhibit grotesque external symptoms. My observations consistently demonstrated that the covering of thick body hair brought on by hypertrichosis is interrupted by patches of epidermal necrosis and inflammation brought on by the HCV. Depending on the severity of HCV infection, one may also observe regions of highly decayed flesh, even exposed bone, but even these sites may exhibit reticulated scar tissue as the enhanced healing rate brought on by LPV attempts to overcome the damage. I have also observed that the enhanced lichen sensory abilities are often moderately reduced in these late-stage victims. Interestingly, however, olfaction may actually be improved, a noted side effect of HCV's proliferation of olfactory receptors and limbic neurons. Ultimately, though the LPV attempts to resist and counteract any new infection, even one so aggressively damaging as HCV, prognosis for the late-stage host is highly dependent on initial overall health and viral load. In my testing of the total sample size were able to resist the long-term effects of HCV effectively, exhibiting only moderate symptoms before returning to near baseline lichen health levels. In individuals, however, symptoms were more pronounced, and limited tissue necrosis, reduced cognition, and sensory impairment persisted for the duration of the study. Long-term observations will be required to accurately determine the extent of this variation, especially in relation to longevity. But while the preceding two phases provided invaluable data, neither could prepare me for the horrors that awaited me in the third. At this point, I must admit the psychological toll of this testing was beginning to wear not only on me, but also my assistant. These are cruel diseases, and frankly, after watching person after person descend into this madness and pain, continuing becomes a matter of pure will. Now, let's see. In any case, for this third phase of testing, we were able to obtain 3 additional specimens, each in the same age range as the others had been, and between 15 to 20 days post-LPV infection. This is a period of the most profound changes in the lichen morphology. Bones are elongated, the posture becomes stooped, and the victims are in clear and near constant discomfort as nearly every tissue in their body is being actively reshaped. It is this stage that, upon introduction of the human zombic virus, results in the highest level of variability. A moderate dose of HZV was given to each specimen. Approximately 4 hours later, 2 of the 3 specimens began to exhibit hallmark signs of infection, such as a spike in heart rate and the onset of a high fever. Shortly after this, each succumbed to the typical zombic coma, wherein the heart rate slows and eventually stops, while the fever continues, escalating to the point of damaged brain function. The third specimen exhibited a similar initial reaction, but did not fall unconscious, and in fact appeared to recover fully within 3 hours. Of the 2 that did enter the coma, 1 awakened within 2 hours, much shorter than the typical 4 to 6. And though his heart rate slowed significantly, interestingly, it never fully arrested. Sadly, the other specimen did not reawaken at all, and expired within approximately 57 minutes after falling unconscious. An autopsy revealed signs of severe metabolic exhaustion, with decreased ATP production and elevated markers of oxidative stress. Ultimately, the struggle to accommodate and fight the dual viral infections likely contributed to an overall weakened condition and impaired regenerative capabilities. In terms of immune response, this unfortunate outcome is not surprising, as upon co-infection, the immune system faces 2 vastly different pathogens. As previously established, LPV subtly alters host cellular functions without eliciting direct cytolysis, while HZV's replication mechanism is characterized by widespread cell death. In any case, it is clear that the body struggles greatly to cope with these infections, and to be honest, it is astounding that any individuals survive at all. Those that do show just how highly variable and volatile the nature of a mid-stage co-infection of this sort can be. Over the following days, the remaining specimens, A and B as I will call them, exhibited numerous additional and increasingly bizarre symptoms. Externally, specimen A in particular began to exhibit striking asymmetry, with much of the right side of the body showing the enhanced muscular development characteristic of LPV-induced anabolism, while the majority of the left side displayed muscular wasting and necrotic lesions, indicative of HZV's catabolic and cytopathic effects. At the cellular level, this asymmetry appears to be the result of localized viral dominance, wherein LPV's integration into the host DNA in some bodily regions promotes protein synthesis and muscle fiber hypertrophy. HZV's replication in other regions leads to increased proteolysis and apoptosis of muscle cells. Additionally, the skin and fur exhibited abnormalities ranging from robust fur growth to ulcerated necrotic patches. Specimen B, which did not enter the zombic coma, exhibited a far less pronounced juxtaposition. However, signs of severe cognitive decline became rapidly apparent. In typical lichens, this transformative stage is marked by increased aggression and an apparent loss of higher reasoning, largely resulting from the reduction of the prefrontal cortex. However, despite this, they often remain cunning, showing signs of lower intelligence, particularly in their hunting and evasion strategies. Specimen B's behavior, however, degraded further, He often flung himself against the walls of his containment, even to the point of personal injury, and regarded us with vicious, mindless intensity. In truth, it was a tragic thing to watch, and it resurfaced memories of finding Allison's body, so mangled and broken. In the specimen before us, the HZV began to degrade the senses of sight and hearing, and the characteristic lichen responsiveness was lost almost entirely, with the notable exception of smell. Interestingly, the eyes of specimen B exhibited a striking heterogeneity. The left eye retained the enhanced clarity typical of lichen transformation, while the right eye exhibited cloudiness and reduced responsiveness, a sign of HZV-induced cataract formation. Within 24 hours following exposure to HZV, both specimens exhibited what at first appeared to be improved reflexes. I attributed this to the combined regenerative effects of the viruses, particularly to the increased muscular innervation caused by HZV. However, the specimens soon exhibited overly heightened sensitivity to stimuli, to the point that gentle prodding seemed to induce visceral reactions, as well as sporadic muscle spasms throughout the body. It became apparent that what we were seeing was, in essence, a zombified brain barely in control of a hulk of muscle and teeth. Particularly in specimen A, heart rate never returned to pre-coma baseline levels, and blood samples indicated increasingly viscous consistency due to raised iron and bile levels, which in turn reduced overall circulation. This led inevitably to a continual spread of necrosis in multiple tissues, particularly of the muscle and surface dermal layers in the extremities. However, because the heart continues to beat, unlike in single HCV infections, contractions of the skeletal muscles are not critical to circulation. In one way, this is good, because the muscular spasms in co-infected individuals appear random, uncoordinated, and somewhat violent, a far cry from the systemic, almost rhythmic pulsations seen in single infected. Specimen B exhibited far fewer spasmodic contractions, especially as the lichen transformation progressed. In this state of partial transformation, however, caught in limbo between two warring pathogens and in nearly constant distress, both specimens tragically died within 2 weeks following co-infection. Indeed, prognosis for organisms affected by this particular co-infection is dire, to say the least. Even on their own, each strain greatly increases morbidity, LPV due to the immense physiological toll such a transformation takes on its host, even if they survive past that point, and HZV due to its inherently destructive nature. Still, a traditional LPV victim may live for years in the lycanthropic state. However, with the exception of the late-stage victims, all of the specimens I observed were deceased within 3 months. Interestingly though, even for those that did not survive, death was preceded by a general period wherein the victims appeared to stabilize, perhaps having reached a kind of equilibrium between LPV's regenerative effects and the degenerative effects of HZV. However, it quickly became apparent that this continuous tension, compounded by the energy and resource demands of fighting off 2 viral infections, would simply deplete the host's physiological reserves. One aspect of my work that will require more in-depth studies is that of transmission. Given our small sample size, which only included infected specimens, I was unable to determine whether the rate of infection increased as a result of co-infection, particularly through the preferred transmission vector of both contagions, a bite. Of course, a co-infected individual would likely exhibit unpredictable transmission characteristics. For example, in the wild, the increased aggression and physical capabilities of a kind of lichen-zombie hybrid may make them more effective at spreading both infections. However, the clear and visible signs of decay and the erratic behavior associated with this co-infection could make potential victims more cautious and less likely to engage with the host, potentially reducing transmission opportunities. It also appears that the loss of the cunning lower intelligence in the lichen brain makes them far less efficient hunters, and therefore less likely to be able to spread either disease. By the time my study concluded, my perspective on these viruses and their effects on a host were forever altered. Beyond the biological effects of each individual strain, the complete usurpation of the victim's humanity is staggering. For now, their prevalence in the wild is minimal, especially in humans, and outbreaks have been swiftly contained. But what will happen if these infections begin to overlap more frequently? I fear the result is much greater difficulty in containment. A zombified lichen, after all, a creature whose outward appearance is a patchwork of symptoms, possesses only a singular, mindless directive: to consume at any cost. I have described the virus that leads to the grotesque twisting of the human form into a vampiric predator, living but profoundly altered. I have also described a pathogen that reduces its host to a shambling, decaying husk. Taken on their own, each condition appears quite different, and indeed, while one enhances its host to near superhuman abilities, the other degrades them to a point that they are little more than a walking corpse. In order to understand both the human vampiric virus and the human zombism virus more fully, I knew that after documenting their individual effects, the next logical step was to document their interactions with each other. Now, my valued listener, I will describe my findings in detail, but I must warn you, the ultimate result of this degree of co-infection is, to borrow a phrase, not for the faint of heart. These insidious infections burrow their way into nearly every cell in their host's body, leaving in their wake a creature that is neither human, nor fully vampire, nor fully zombie, but something far more terrifying. While my discovery of the notable zombic pathogen occurred relatively early in my career, the personal journey that led to the discovery and subsequent documentation of the virus that causes vampirism was long and destructive. I have documented many creatures and pathogens previously unknown to science, and though many will likely never become public knowledge, the work that my team and I did, is of vital importance. If the average person understood the true depth of the biological threats that lurk just beyond their awareness, well, methods like those I must employ might just seem justified. At least, that is what I tell myself. In any case, utilizing the same remote, abandoned medical facility in which we conducted our previous studies of co-infected individuals, my research assistant and I once again established secure containment for any participants in this next phase. Securing willing participants for infection with a vampiric virus was surprisingly easy. For a certain demographic, all that was necessary was to present the potential effects, and our pool of willing participants grew easily. In fact, even the possibility of the introduction of the zombic virus did little to dampen some individuals' enthusiasm. And so it was that our study commenced. Now, before I proceed, and though both have been explored in great detail previously, it is worth our time to briefly present the details of both pathogens here, beginning with the human vampiric virus, or HVV. HVV is a member of the mononegavirales order and is an RNA virus that targets a wide range of human cells, with the exception of red blood cells. Upon entry, the virus utilizes a unique strategy, infecting cells through endocytosis and altering their function to produce viral clones, without causing immediate cell destruction or lysis. One of the most striking features of HVV is its rapid replication cycle, facilitated by the infection of the thyroid gland and the subsequent increase in metabolic processes throughout the body. This, coupled with an incubation period of a mere 6 to 12 hours, allows the virus to quickly establish itself within the host. As the infection progresses, the host experiences a range of symptoms, from initial flu-like manifestations to the onset of the vampiric coma, a state of active unconsciousness during which the body undergoes a dramatic transformation. This metamorphosis includes elongated canines, enhanced sensory abilities, and a heightened capacity for regeneration, among other changes. But the changes themselves are not entirely for the better. The virus also rewires the host's brain, leading to increased aggression, sensitivity to light, and an insatiable thirst for blood. The human zombism virus is also a member of the mononegavirales order, and it shares some of HVV's fundamental characteristics. Like HVV, HZV is a single-stranded RNA virus that targets a wide range of human cells. However, HZV's replication strategy differs significantly. While HVV relies on endocytosis and the alteration of host cell function to produce viral clones, HZV utilizes a reverse transcriptase enzyme to produce DNA from its RNA genome. This DNA is then integrated into the host's genome through an integrase enzyme, effectively transforming the infected cell into a factory for new HZV virions. The consequences of this replication strategy are severe, leading to widespread cellular damage, necrosis, and a rapid decline in the host's overall physiological condition. And rather than the relatively slow progression observed in HVV victims, in HZV, symptoms such as aggression, cognitive decline, and physical decay manifest within hours of exposure. Another key difference between the two is the nature of the zombic coma observed in HZV. Unlike the vampiric coma, which is characterized by active unconsciousness and metamorphosis, so to speak, the zombic coma is a state of near-complete metabolic shutdown that mimics death. This coma lasts between 4 to 6 hours and is followed by a reanimation phase wherein the individual reawakens with enhanced innervation of skeletal muscle, but a complete loss of humanity. Now, as I briefly touched on in a previous recording, coinfection is a phenomenon in which a host is simultaneously infected with 2 or more distinct pathogens. In the realm of virology, co-infections can involve viruses from the same family or from entirely different families, each with their own unique characteristics and pathogenic mechanisms. In short, when 2 or more viruses infect the same host, they may interact in complex ways, leading to synergistic or antagonistic effects on viral replication, immune responses, and disease severity. For this study, I thought it best to begin with 2 groups, a control group, which were allowed to progress through the stages of HVV infection without intervention, and an experimental group, which were exposed to HZV at a predetermined point before the onset of the vampiric coma. Under strict biocontainment protocols, we introduced HZV to the experimental group via controlled exposure. The subjects were then closely monitored for signs of co-infection, with regular assessments of their vital signs, cognitive function, and physiological state. The results were both fascinating and disturbing. Within a mere 2 hours of HZV exposure, the experimental group began to exhibit symptoms that deviated sharply from the expected course of early-stage HVV infection. As in normal HVV infection, subjects exhibited rapid onset of high fever, accompanied by severe chills, profuse sweating, and a marked increase in heart rate, some up to 150 beats per minute. Moreover, subjects reported severe thirst despite consuming water at a rate nearly 3 times higher than the control group. This, of course, was paired with frequent urination, which suggested the rapid onset of metabolic imbalances and potential kidney dysfunction. Cognitive impairment followed rapidly, with many subjects experiencing severe confusion, disorientation, and memory loss, with some unable to recognize their surroundings or recall basic personal information. This level of pre-coma cognitive decline was not observed in either the control group or in our previous studies of HVV or HZV infections alone. And in fact, as the co-infection progressed, the increased metabolic activity brought on by the initial HVV infection actually appeared to accelerate the spread of HZV throughout the body. This vicious cycle of viral replication and cellular destruction led to a universally rapid deterioration of the subject's condition. Many developed severe respiratory distress, with oxygen saturation levels dropping below 90%, while neurological symptoms such as seizures and hallucinations became more frequent and intense. Many of the subject's skin took on a mottled, grayish appearance, and numerous lesions and necrotic patches began to develop. But of course, a hallmark of both infections is the rapidly induced comatose state, in HVV beginning 12 to 24 hours after initial exposure, and 6 to 12 hours in HZV. This state is induced in response to overwhelming metabolic stress. In an effort to cope with the numerous transformative processes brought on by the respective virus, the body's only recourse appears to be to shut down all non-essential functions, including consciousness. Duration of the coma is typically 24 hours and 4 to 6 hours, respectively. All 10 subjects in the experimental group entered into a comatose state within 10 hours of HZV exposure. This coma, which I have termed the hybrid coma, exhibited characteristics of both viruses and was characterized by a profound loss of consciousness, accompanied by a significant decrease in metabolic activity and a slowing of vital signs. Subjects regained consciousness, or some semblance thereof, within an average of 18 hours. Upon emergence from the hybrid coma, subjects exhibited an unprecedented range of symptoms and outcomes. 7 individuals exhibited an apparently accelerated progression of both vampiric and zombic traits, with the rapid onset of enhanced sensory abilities and increased strength. Simultaneously, however, they began to show signs of cognitive impairment and a rapidly increasing aggression toward any human in the near vicinity that far outpaced the initial disorientation of HCV or the more gradual hunger of HVV. The final outcome, at least for 3 specimens, was death. These subjects did not awaken from the coma, instead succumbing to what appeared to be overwhelming systemic failure. The precise cause of this sudden deterioration remains unclear, but at this time, it seems likely to simply be the result of the breakdown of multiple homeostatic mechanisms in response to the competing infections. Of course, observations continued for some time, and the results became even more intriguing. Remarkably, 3 of the remaining individuals demonstrated a partial resistance to the zombic aspects of HCV. They displayed enhanced physical capabilities and sensory acuity typical of HVV infection, but the expected cognitive decline and necrotic tissue damage associated with HCV were notably absent or significantly delayed. This unexpected resistance appears to have been the result of an unusually robust immune response, as evidenced by the presence of a high number of antiviral cytokines in the specimen's blood. This suggests that while the subject would still suffer from the effects of HVV, the most lethal aspects of HZV might be mitigated. Lifespan in these individuals has so far supported this. Continued observation will be necessary to determine for certain, but it stands to reason that these 3 individuals may survive for greater than a year. Moving along, 2 subjects entered a state I have termed unstable hybridization, wherein the subjects oscillated between the behavioral and physical characteristics of their respective infections. Periods of lucidity and enhanced physicality were abruptly interrupted by bouts of aggressive, zombic behavior and visible physical decay. As you might have guessed, these fluctuations posed considerable challenges to containment and care. The precise cause of this oscillation isn't immediately apparent, but it may be driven by episodic changes in the host's metabolic state and influenced by factors such as pre-existing levels of stress, nutrition levels, and circadian rhythms, which may have intermittently favored, for lack of a better word, one virus over the other. This oscillation results in the erratic expression of viral genes, leading to the observed phenotypic instability, as well as significant physiological stress and, over time, systemic damage. Ultimately, after emerging from the hybrid coma, these subjects were deceased within an average of 32 days. 2 other subjects experienced rapid physical deterioration unlike any observed in the control groups. Their bodies in particular seemed to more deeply manifest the internal struggle of the competing infections, resulting in an accelerated cycle of tissue regeneration and necrosis. Subjects exhibited grotesque physical abnormalities, including rapidly fluctuating muscle mass, erratic bone growths, and extensive dermal lesions, the severity of which seems to indicate an overactivation of certain signaling pathways involved in cell growth and death, such as those associated with HVV-induced regeneration and those associated with HZV-induced inflammation and necrosis. The simultaneous activation of these pathways likely leads to conflicting signals within cells, causing chaotic cellular behavior and tissue pathology. It should come as no surprise that this severe physical and metabolic stress reduced lifespan to approximately 4 days post-coma. The final subject presented a unique case of what could only be described as advanced hybridization. This individual displayed an unprecedented integration of both viral traits, with high physical and sensory enhancements, with minimal cognitive decline, and what appeared to be controlled necrotic progression. Intriguingly, this subject managed to maintain a level of self-awareness and could communicate effectively, albeit with noticeable difficulty. At the time, I believed this to be nothing more than a novel form of viral symbiosis, wherein HVV's enhancements compensated for HZV's degenerative effects, essentially leading to a state of relative balance and allowing the subject to function semi-normally. While there was certainly some truth to this, it wasn't until Phase 3 that I would begin to truly understand what was happening here, at even the genetic level. But I'm getting ahead of myself. We will return to this discussion in due time. For now, allow me to provide the record of what came next. The second phase of our study was designed to observe the effects of zombic infection in subjects with established vampiric traits, specifically those who had already undergone the vampiric coma and emerged as fully transformed vampires. Test subjects were easily recruited from the previous control group, who were infected for at least 6 months and had fully adapted to their new vampiric physiology. I should note here that while, of course, locating these individuals was a matter of ease, persuading them to join in this effort was another thing altogether. In any case, as before, subjects were divided into 2 groups, a control group, which would continue to live as vampires without any intervention, and an experimental group, which would be exposed to the human zombism virus. Within the first 24 hours of HZV exposure, the experimental group began to exhibit symptoms that were distinct from both their established vampiric baseline and the typical progression of HZV infection in humans. And the results, as should likely be expected by now, were both intriguing and disturbing. One of the most striking initial symptoms in these subjects was a rapid onset of physical pain. Subjects reported intense burning sensations throughout their bodies, particularly in their small muscle groups and joints. This pain was apparently so severe that many were unable to move or even stand, a stark contrast to the usual vampiric strength and agility. Now, in traditional zombic infections, as mentioned in my direct study, it is unlikely that pain is perceived strongly. As the senses degrade, it is likely that victims of this infection feel a chronic aching sensation, but the loss of higher brain function limits the interpretation of severe pain signals. The effects of the vampiric virus, however, counteract much of this nerve blunting and appear to prolong higher brain function. As a result, the subjects in this group experienced severe discomfort likely stemming from joint inflammation, myositis, nerve tissue destruction, and various necrotic processes. Because of this, several subjects were at last sedated in an attempt to relieve their suffering. But this discomfort was only exacerbated by profound hunger and thirst. Despite having fed on blood just prior to HZV exposure, each subject reported an insatiable craving for both blood and flesh, with no apparent distinctive preference. Indeed, it appears that HZV-induced damage to the hypothalamus disrupts the process of hunger signaling, leading to an indiscriminate feeding pattern, a far cry from the selective sanguivary of traditional vampires. Worse, this indiscrimination is paired with the frenzied aggression observed in zombies, entirely devoid of the calculated hunting strategies seen in vampirism. Autopsies revealed another possible explanation for this altered feeding pattern. The stomach and intestines exhibited severe necrosis, which presumably limited the subject's ability to process blood effectively. As the infection progressed, and as with the previous phase, the subject's skin, which had previously been pale and smooth, took on a grayish, mottled appearance. Necrotic lesions and patches of decaying flesh began to appear, particularly around the site of HCV exposure. Cognitive and behavioral changes were equally concerning. Subjects routinely exhibited periods of extreme confusion, disorientation, and memory loss, struggling to recall even basic information. These cognitive impairments were punctuated by bouts of intense aggression and rage, with subjects lashing out at both their surroundings and each other. In terms of senses, in 92% of the experimental group, visual acuity was substantially diminished as ocular tissues were compromised by HZV, and the same was true of hearing, though the sense of smell appeared to actually surpass that of the control group. Blood samples and tissue biopsies taken from the experimental group indicated an increasingly higher viral load of HZV relative to HVV over time. This seems to indicate that HZV's more aggressive infection strategy is superior, at least in terms of replication, than that of HVV, and in short, the zombic virus appeared to disrupt the delicate balance HVV had achieved in maintaining its own dominance within the victim. Even so, the established presence of HVV within the subject's cells did appear to modulate the progression of HZV infection in some unique ways. For example, rather than inducing lysis in all cases, the presence of HZV in the cells more commonly resulted in a state of cellular dysregulation. It appears that mitochondrial function, which HVV had enhanced in order to support the vampiric physiology, began to deteriorate, leading to decreased energy production and increased oxidative stress. At the tissue level, this cellular dysregulation manifested as a kind of tension between regeneration and decay. Some areas of the subject's bodies, particularly those with a high concentration of HVV-infected cells, appeared to resist the destructive effects of HZV, maintaining a semblance of vampiric integrity. Other areas, however, succumbed to the aggressive spread of HZV. I suspect this variance to be the result of viral tropism, as well as the varying strengths of localized immune responses in the varying tissues. Due to the compounded physiological strain and systemic breakdown caused by the two viruses' competing mechanisms, the life expectancy of subjects co-infected with HVV and HZV is drastically reduced, ranging from a few weeks to several months post-infection, and significantly shorter than what might be expected from an HVV infection alone. The final cause of death in these subjects was accelerated organ failure and eventual complete systemic collapse. While the effects observed in the first two phases were equal parts distressing and enlightening, a good portion of what we documented was, in a way, expected. But in the process of our observations, and as our sample sizes grew, an entirely unexpected result began to appear more and more frequently. I alluded to it briefly in my recounting of the first phase, but little did I know then just how insidious this particular co-infection would prove to be. I must take a brief intermission, my valued listener, but when I return, I will explain these findings in detail. For now, suffice it to say that what this co-infection produced was, I believe, a novel form of viral evolution, and it was occurring right before our eyes. I've grown accustomed to experiencing what many outside this field might consider horror. In truth, there is no evil to be found in nature. Though it can be vicious and even frightening, there is wonder in the chaos, a beauty even in the brutality. But sometimes the beauty can be difficult to find, obscured as it is by our basest fears. Nowhere has this dichotomy been more apparent than in my studies of pathogens, many of which transform their hosts into twisted shells of humanity, if any humanity remains at all. Now allow me to continue my documentation of one such study, the interaction between the human vampiric virus and the human zombism virus in a co-infected individual. While these certainly provided invaluable data on their own, it wasn't until the third phase that we observed the most altered progression of symptoms. Indeed, despite my attempts to dispel it, during this phase, only one word could capture what the results elicited in us: horror. In the experimental groups of both prior phases, we began to observe what I considered to be an atypical progression of symptoms, ones that did not align with the established models of either virus. First was the unusual blend of characteristics observed in many subjects, such as the rapid onset of necrosis coupled with the regenerative effects of HVV. This often led to accelerated healing in areas adjacent to necrotic tissue, something I thought unlikely to be explained simply by the additive effects of co-infection. Second, it appeared that many co-infected subjects exhibited unusual immunological responses. For example, the antibodies we normally associate with each respective virus were present, but more commonly, we observed an additional antibody response that appeared to target unique viral epitopes, ones that are not present in either virus alone. I decided to pursue additional genetic testing performed on the viral RNA extracted from tissue samples of co-infected subjects. Astoundingly, sequencing data revealed viral genomes that contained genetic elements from both HVV and HZV. It is my hypothesis that what we were witnessing was, in fact, a form of recombination. As you may already be aware, recombination is a process by which genetic material from two or more different sources is combined to create a new hybrid genome. In the context of viruses, Recombination can occur when two or more distinct viruses infect the same host cell and during the replication process exchange genetic material. Because both HZV and HVV share a classification within the Mononegavirales order, it should have been clear that this was a possibility. Instead, the genetic similarities between the two viruses, coupled with their ability to infect the same host cells, were the perfect combination for the emergence of a new recombinant pathogen. Though much more testing will be necessary to determine the exact process, it is my hypothesis that during viral replication, the RDRP enzyme, itself responsible for synthesizing new viral RNA, may have switched templates from one viral genome to another, resulting in the incorporation of genetic material from both viruses into a single chimeric RNA strand, a process known as copy choice recombination. In short, the resulting recombinant virus, which until a more proper name can be determined I have simply called the human vampiric zombism virus, or HVZV, displays a unique combination of genetic elements and functional properties derived from both parent viruses. And though the specific genetic composition of HVZV is likely varied among different recombinant strains, there are some notable consistencies. At the cellular level, once inside the host, HVZV's chimeric replication machinery appears to allow for extraordinarily rapid viral proliferation and spread. The recombinant virus also demonstrates an increased ability to evade host immune responses, primarily due to the acquisition of immunomodulatory genetic elements from both parents. Once the recombinant virus becomes more prevalent, the next phase of our study was clear. We needed a group of subjects who hadn't been exposed to either virus and would only be infected by the novel HVZV. Once more, we obtained the willing participation of a handful of individuals, pioneers really, who would undergo this unprecedented trial. And it was in this group that we observed the most profound changes yet. The rate of infection in this strain is staggering. While the incubation period averages about 3.5 hours, the expected comatose state was reduced to a state of altered consciousness, more like delirious catatonia. This period routinely spanned 2 to 4 hours. Following this, the virus had spread to nearly every tissue within a matter of hours and had already begun to affect its changes. Internally, in almost all of these individuals, the remodeling that occurred in HVV still took place, but in novel ways. For example, during the coma, the heart ceases to function, presumably due to viral cardiomyopathy, and it appears that it will eventually necrotize completely. As in both HZV and HVV, the skeletal muscle will take over the function of maintaining circulation through rhythmic contractions. In HVZV, these contractions are more controlled than those seen in zombies, resulting in improved circulation, which in turn limits the spread of necrosis. However, these contractions are occasionally spasmodic, causing the subject to tense completely in clear discomfort. The muscles themselves exhibit the proliferation of fast-twitch fibers characteristic of HVV infection, while also recruiting additional muscle fibers in the forearms, hands, and jaws. This results in a subject with the ability to not only move quickly, but exert astonishing grip and bite force. However, I have not observed a significant increase in myoglobin or mitochondrial efficiency, which would explain why, despite being capable of large bursts of speed and power, HVZV victims exhaust relatively rapidly. Now, in a zombic infection, while bite force is also greatly increased, the teeth are not reinforced, leading to their eventual destruction. It appears, however, that during the genetic recombination of HVZV, the vampiric virus's osteogenic and dentinogenic effects that normally apply only to the eye teeth have been somehow modified to include the incisors and, to a lesser extent, premolars. This will certainly require more study to understand fully, but the effect of this change is clear. These modified teeth are a disturbing adaptation for its altered dietary habits. In short, HVZV-infected hosts exhibit a compromised digestive system, which appears to struggle to process and extract nutrients efficiently. Behavioral observations indicate, as in the second phase, that feeding is indiscriminate. And yet, thanks to periodic necrosis of the gastrointestinal lining, The digestive system is not completely adapted to the processing of blood, leading to apparently severe pain and impaired nutrient absorption. Even so, nutrients are absorbed, but the rate of feeding must increase to compensate for its inefficiency. Furthermore, HVZV victims appear to lack the ability to synthesize PCDH-Y, just as in HVV, and must obtain it from their food. As a result of this, recombinant victims are deeply aggressive. Indeed, it appears that the wild frenzy of the zombie is paired with the relatively controlled desperation of the vampire, leading to an extremely effective killing machine, one who is able to hunt with ruthless precision. Despite some level of control, however, it is unlikely that anyone exhibiting this affliction would ever be able to blend into society, as many vampires attempt to do. As I've often stated, the nature of these studies and their associated observations, despite their importance, can take a heavy toll. I consider myself, and even my assistant, to be of relatively stout constitution. It may sound unscientific, but in fact, in watching the subjects as these diseases wrought their insidious and often painful changes, it was clear that anyone who feels nothing at the anguish of another could certainly not be trusted to conduct an experiment objectively. It is my sincere hope that these tests will soon come to an end, and the era of such pernicious pathogens can come to a close. But beyond the physical discomfort exhibited by most of the subjects in this study, it was the changes in cognition that gave me the most pause. Indeed, cognitive functions are profoundly impacted by HVZV. Likely due to the influence of HVV, overall subjects appear to retain a semblance of their pre-infection personalities. However, this is often interspersed with memory lapses, disorientation, and a temporary loss of higher reasoning skills. Similarly, emotional regulation is severely compromised, and infected individuals are highly dangerous and unpredictable. Periods of emotional stability are rare and fleeting, often giving way to intense outbursts of violence. And yet, it is when a prolonged moment of lucidity intersects with awareness of their condition that the most disturbing emotion appears: despair. Fortunately for the victim, though a semblance of balance is maintained for a time, eventually their higher cognitive functions will devolve beyond even this fleeting awareness. The cause of this degradation of cognition appears to be the disruption of synaptic function, in turn caused by the direct infection of the recombinant virus within neuronal cells. The resulting erratic nerve function affects much of the victim's behavior, but interestingly, it does not, at least initially, appear to affect motor control. As in the progenitor HVV, victims of HVZV exhibit enhanced nerve conduction velocity, as well as thicker motor nerve axons, both of which lend to incredibly fast and precise movements and reflexes. As in zombies, I have observed massive stem cell production and the noted absence of glial scarring, both of which lend to astonishing regeneration abilities in damaged nerve cells. In short, if any normal human being were to find themselves stalked by a victim of HVZV, little short of complete destruction could possibly succeed in stopping an attack. But although attacks are powerful and destructive in nearly every case, in HVZV victims, the senses that normally assist with discerning and locating prey are difficult to quantify. For example, the gustatory cortex in many individuals appears to be enlarged, implying the enhanced sensitivity of taste. This, however, is paired with an apparently strong preference for the tastes of blood and living flesh, with varying ability to detect and interpret any other flavor. And as oral tissues deteriorate, taste sensitivity appears to fluctuate rapidly, which only adds to the infected's confusion. Hearing is another sense that is difficult to analyze. It appears that while HVZV victims exhibit enhanced abilities to detect even minute sounds at a distance, some even being drawn to the sound of a beating heart, it seems difficult for the same individuals to locate the sound's source. Indeed, it appears that in each case, asymmetrical enhancements lead to inconsistent sound localization and directional hearing abilities. Olfaction, on the other hand, is greatly increased in effectiveness, as olfactory bulbs and associated cortical processing areas appear hypertrophied in HVZV victims. Though field testing in this study was limited, it can be safely concluded that the scent detection ability in these individuals surpasses that of both vampires and zombies. A truly frightening possibility. The most unpredictable outcome of all, however, had to do with sight. In HVV, victims exhibit quadrochromatic vision, with an additional fourth cone type being tuned to near-infrared. This also appears to occur in victims of HVZV, as does the characteristic formation of tapetum lucidum, which greatly enhances night vision. However, these visual improvements are paired with sluggish pupillary response and reduced function in the visual cortex of the brain, both of which are owed to the influence of HZV. Cataract formation occurred in roughly 40% of individuals as well. The result is a creature with the ability to see a broad spectrum of light and can see well in near darkness, but has difficulty discerning silhouettes and cannot cope well with rapid changes in lighting conditions. Once again, this affects these individuals' ability to hunt with precision, having the counterintuitive effect of making them more dangerous. Interestingly, these deteriorative changes also appear to have reduced the intense reaction produced by intersecting right angles, a trait normally observed in vampires and which I have previously described as the crucifix glitch. Indeed, subjects presented with a cross displayed no adverse reactions, much less the typical seizure-like neuroelectric overload observed in vampires. Lastly, as we observed in the second phase of our study, individuals afflicted by the recombinant virus began to experience chronic inflammation. This, combined with the virus's aggressive replication within nerve tissues, appeared to amplify pain perception. Indeed, even minor injuries seemed to elicit severe reactions, and in 83% of those studied, subjects expressed chronic levels of severe discomfort. This appears to have been alleviated for a time by the influx of dopamine associated with feeding. It is perhaps fortunate for the infected that, given the number of maladaptive traits brought on by this particular virus, their expected lifespan is drastically reduced from what we would expect to see in vampiric individuals. Indeed, it appears that the physiological stress, combined with chronic inflammation and frequent tissue damage, will eventually lead to the failure of vital systems. But more than that, it appears that because of the increased cell turnover resulting from continuous cycles of tissue damage and repair, HVZV actually accelerates telomeric erosion. This occurs so rapidly, in fact, that the increased levels of telomerase brought on by HVV cannot effectively counteract it. The ultimate result is that individuals afflicted with HVZV will likely not exhibit the lengthened lifespan of vampires, but nor do they succumb to the necrotic effects of HZV as rapidly as true zombies. Instead, they are caught in a kind of horrific limbo, highly dangerous, yet equally impaired, their minds and bodies degrading enough to cause pain and unending hunger, yet functional and aware enough to recognize the slipping away of who they once were. If you've listened to this recording to this point, my valued listener, I believe it highly likely that you consider me to be as much of a monster as the poor creatures I've studied. And in some ways, perhaps you're right, but this work must be performed. There's simply no other recourse. This mantle has been thrust upon me, and if the civilized world is to remain blissfully unaware of the threats that face it daily, I could no more refuse it than I could the offer that ushered me into this world of shadow in the first place. My apologies. At this point, you may be wondering, even as I did, given the apparently close relationship of HVV and HZV, and despite their distinct pathologies, is it possible that they descend from a common ancestor? Though I have no direct evidence yet, it is my belief that the answer is yes. Based on the similarities in their genomic structure, cellular entry mechanisms, and zoonotic origins, I propose the existence of a hypothetical progenitor virus, which I have tentatively named the hematophagic mononegavirus, or HMV. The HMV, I believe, originated in an ancient blood-feeding organism, likely an insect or arachnid parasite. The virus would have developed the ability to persist in the host's bloodstream and to be transmitted through the vector's feeding process. Over time, the HMV could have adapted to infect multiple species, including bats and other mammals, through the bites of infected vectors. In these intermediate hosts, it seems likely that the virus underwent further mutations and adaptations, allowing it to better exploit the host's cellular machinery for replication. As the HMV spread to different host species, it likely encountered various unique pressures, possibly in the form of immune responses and competition with other microbes. These pressures could have driven the divergence of HMV into distinct lineages, eventually giving rise to the early form of the human vampiric virus and the human zombism virus. The next step in this research is twofold. First, epidemiological investigations into the geographical distribution and transmission patterns of HVV and HZV, which could potentially reveal common hotspots or overlapping regions where the viruses first emerged. Second, more in-depth genomic sequencing and comparative analyses of HVV and HZV isolates from different hosts and regions, perhaps even non-humans. These could identify shared genetic elements and conserved molecular features, each of which could allude to a common ancestral virus. Until such a time as these additional studies can be conducted, it is difficult to even speculate as to the nature of this progenitor virus in relation to its hosts. It is likely that victims of HMV exhibited acute fever and flu-like symptoms, alterations in behavior such as increased aggression or agitation, heightened senses, especially those related to the detection of blood or potential prey, increased metabolic rate and energy consumption, and perhaps a gradual deterioration of cognitive functions. However, the specific symptoms and their severity would likely vary greatly depending on the host species and the stage of viral evolution. It would be irresponsible of me to confirm any specifics at this time, but suffice it to say that I have wondered if perhaps some of the worldwide legends of the undead could point to a single source, a gap in scientific knowledge that HMV could potentially fill. Ultimately, it has become clear to me that given this recombinant virus's enhanced rate of infection, it is now more important than ever that both HZV and HVV be eradicated, or some kind of cure developed. Even this study was brought to what I consider to be a premature end, as the risks associated with this novel pathogen far outweighed the potential benefits of continued research. The decision to halt the experiment was not made lightly, but the possibility of an unintended release was simply too great. It is my hope that similar studies will be conducted in the future, but with resources and safety measures to which I no longer have access. I will admit that there are times, more frequently now, when I take no joy in my work. There has been intrigue and passion, but the weight of bearing this knowledge has begun to take a toll. The world is still full of mystery, but lately I've begun to wonder if my studies will truly dispel the darkness that conceals them, or if that darkness will simply swallow us whole. But now, my valued listener, I must present to you a new phenomenon, one so disturbing that I hesitated to even bring it about for observation: the co-infection of HVV and LPV in a single host. In this study, I will detail my observations of individuals unfortunate enough to fall victim to both viruses, neither fully vampire nor completely werewolf, but something else. I only ask that you withhold judgment as to my methods. In a world where such pernicious diseases not only exist, but seem to be spreading more rapidly than at any point in human history, some sacrifices cannot be avoided. We have discussed the individual effects of both HVV and LPV in previous volumes, but in the interest of thoroughness, allow me a moment to summarize each disease respectively. The human vampiric virus is a member of the order Mononegavirales. It penetrates nearly every cell in the human body, sparing only the red blood cells, acting swiftly and largely silently. Indeed, its speed is remarkable, with victims succumbing to its influence within a mere 6 to 12 hours of infection. Transmission occurs primarily through the exchange of bodily fluids, most commonly through the bite of an infected individual. The hallmark of this virus is the vampiric coma, a day-long period of active unconsciousness during which the host undergoes a disturbing metamorphosis. Upon awakening, the victim's strength and reflexes are enhanced, and their brain essentially rewired, granting quadrochromatic vision that extends into the near-infrared spectrum, but at the trade-off of extreme photosensitivity. Victims of HVV also experience a fundamental shift in metabolism. Due to the effects of the virus, such victims lose the ability to synthesize a crucial protein found only in hominids, and they find themselves driven to consume both human blood and flesh. In exchange, the virus offers its host virtual immortality, continuously regenerating cells, staving off aging for astonishingly long periods of time. The lycanthropic parvovirus also reshapes its victims, but the process is far more prolonged and agonizing. This DNA virus, a distant cousin to the parvovirus that afflicts common household pets, likely began in canines before mutating in a way that allowed it to infect humans, and in doing so brought along a suite of genes that fundamentally alter the human body plan. The initial stages of LPV infection manifest over a day, but this is only the beginning of a roughly 6-week ordeal of excruciating transformation. As part of this horrifying process, bones elongate and reshape, muscles hypertrophy, and the facial structure is entirely altered, eventually coming to resemble a canine muzzle. The virus also triggers widespread hypertrichosis, while the senses, particularly hearing and smell, are deeply enhanced. Shortly after initial infection, higher brain functions begin to deteriorate, leaving the victim a slave to primal instincts and uncontrollable aggression, while circadian rhythms are completely flipped. The result is an aggressive quadrupedal beast that is scarcely recognizable as having once been human. As documented in previous volumes, thanks to a connection of mine somehow unstrained due to the events of the past few years, I managed to obtain access to a decrepit, though isolated and secure, facility for testing. Though it is an advantage to utilize such a facility, I have grown to hate it. My only solace is the necessity of these studies and the hope that it will all soon be over. It took more time than in previous studies to find suitable individuals for observation. This is in part due to the fact that I anticipated that a co-infection of HVV and LPV would take a heavy toll on even the healthiest participants. But also, given the mortality rates of LPV in particular, I knew that we'd need a far larger sample size than normal. Though I believe that such occurrences will only become more frequent, at this time, the natural co-infection of HVV and LPV in a single individual is quite rare. Indeed, it likely requires the alignment of several circumstances, of which I have identified at least 2. First, there is the highly unlikely scenario of simultaneous infection, wherein the unfortunate victim receives bites from both a vampire and an LPV-infected organism, human or otherwise, within a short time frame, typically less than an hour. Second, and more likely, is a sequential infection, wherein a host infected with one virus encounters a carrier of the other before the first virus's transformative process is complete. I made the decision to form the phases of my study around these 2 primary scenarios, beginning with the second. The first stage was the first of many challenges this entire study faced. The potential for extreme suffering and high mortality rates weighed heavily on my conscience, yet the importance of understanding these co-infections compelled me. But you have likely grown weary of my personal musings, so let us proceed. Through our usual channels, over the course of the study we managed to secure a group of 40 subjects, all between the ages of 25 and 40, and of appropriate physical condition. I divided the subjects into 2 equal groups. Group A to be first infected by HVV, followed by the introduction of LPV, and Group B with LPV as the primary infection, followed by HVV. Each group was further subdivided to allow for varying intervals between the primary and secondary infections, ranging from 6 hours to 5 days. The study began with the controlled exposure of Group A to the human vampiric virus. As expected, within 6 to 12 hours, all subjects exhibited the typical early symptoms: high fever, severe thirst, and elevated heart rate. The lycanthropic virus was introduced to the first subgroup at the 12-hour mark, and the interaction between the viruses was immediate and violent. 3 subjects did not survive the first few hours, their bodies unable to withstand the conflict between the viruses, with the cardiovascular system showing catastrophic failure as the heart struggled to adapt. Now, in a traditional HVV infection, the vampiric coma that follows the infection is a state of active unconsciousness. There is clearly an internal turmoil, but it could be construed as a feverish delirium similar to that observed in other, more common infections. In the subjects of this group, however, the vampiric coma was far worse. Their bodies contorted in unnatural ways, muscles spasming beneath the skin, and most alarmingly, several subjects regained brief moments of terrified consciousness during this period before lapsing back into comatose states. The second subgroup, infected with LPV at the 24-hour mark just as they were emerging from the vampiric coma, fared marginally better. The immediate mortality rate was lower, with only one subject expiring in the first few hours. However, the following transformation process, exacerbated by LPV, was no less horrifying. The result was a grotesque exhibition of partially transformed vampires experiencing rapid and agonizing lycanthropic changes. Their elongated, more gracile limbs thickened and twisted, while limited hypertrichosis grew in asymmetrical patterns across the body. The final subgroup, exposed to LPV 5 days after HVV infection, presented yet another variation in outcomes. These subjects, having completed their vampiric transformation, initially appeared resistant to LPV's effects. However, as days passed, these subjects began to exhibit a blend of vampiric and lycanthropic traits. Their once graceful forms became more robust, with increased muscle mass and bone density. Facial features began to elongate slightly, though not to the extent seen in pure lycanthropic transformations. Among all groups, HVV's enhancement of fast-twitch muscle fibers, combined with LPV's stimulation of protein synthesis, resulted in extreme muscle growth. It was obvious, however, that the subject's nervous system struggled to regulate such rapid changes, which led to extended periods of overstimulation. And because the nervous system had not fully adapted to the increased motor control required by this muscle growth, most subjects experienced involuntary spasms, as one might expect to see in traditional HVV infection, though far more pronounced. Finally, metabolically speaking, subjects were in a state of constant demand. Indeed, nutritional requirements fluctuated wildly between subjects and within individuals, creating a sense of perpetual hunger. Some subjects began to experience muscle wasting and signs of internal organ stress when their needs were not met quickly enough. As this phase of the study progressed, it became increasingly clear that the interaction between HVV and LPV was far more complex than even that of our previous studies. Indeed, these viruses seemed to be vying for dominance within the host's body, resulting in a transformation process more prolonged and agonizing than either virus produces alone, and with a disturbingly high mortality rate. By the end of the first week, 40% of Group A were tragically deceased, with the remainder in various states of agonizing transformation. After some time, we began our study of the next group, Group B, which was to encompass an LPV primary infection, followed by that of HVV. As with Group A, we subdivided Group B to allow for varying intervals between infections: 24 hours, 72 hours, and 7 days post-initial LPV exposure. The results were, to put it mildly, chaotic. The first subgroup, exposed to HVV just 24 hours after LPV infection, experienced the highest mortality rate of all test groups. The introduction of HVV at this early stage of lycanthropic transformation simply seemed to create too high a level of physiological stress. Of the 6 subjects in this subgroup, 4 expired within hours of HVV exposure. Autopsy results of these unfortunate subjects revealed, if you'll pardon the informal terminology, a cellular battleground. Observed samples seem to indicate that LPV's initial alterations to the genome created a hostile environment for HVV's typical mode of infection. In response, HVV seemed to adapt. Indeed, we observed an amplified level of virulence which, in turn, led to rapid and widespread cell death, particularly in the liver and kidneys, and which ultimately resulted in total organ failure. The 2 surviving subjects from this subgroup underwent a terrible transformation wherein the gradual reshaping of bones characteristic of LPV infection accelerated dramatically, so much so that fractures within the bone were audible. Most notably, these subjects never entered the vampiric coma. Instead, they remained in a state of agonized consciousness throughout the entire process, their screams eventually devolving into inhuman howls that seemed to echo through the facility for days. The second subgroup, exposed to HVV 72 hours after LPV infection, experienced a higher rate of survival. By this point, the initial stages of the lycanthropic transformation were well underway, with subjects exhibiting increased muscle mass, the beginnings of facial elongation, and patches of thick hair growth. The introduction of HVV at this stage created a visible conflict in the transformation process. In some areas of the body, the lycanthropic changes continued unaltered, while in others, the influence of HVV seemed to overtake it. As was observed in the co-infection of lycanthropy and the zombism virus, this resulted in a bizarrely asymmetrical development. The final subgroup, infected with HVV a full week after LPV exposure, presented yet another variation in outcomes. These subjects, well into their lycanthropic transformation, appeared to resist HVV infection, though it is difficult to ascertain whether this is because LPV outpaces the viral war for resources, so to speak, or whether the changes brought on by HVV were simply lost among the larger changes wrought by the lycanthropic virus. This particular outcome may warrant further testing. By the conclusion of this phase of the study, the survival rate in Group B mirrored that of Group A, with only 60% of subjects surviving. However, the expressions of the co-infection varied wildly between individuals, and regarding consistency of the interaction of these viruses, little has been determined fully. While there were many observable differences in each of the groups, there were many things that I do consider consistent in this variety of co-infection, at least enough to warrant an overview of certain symptoms and outcomes. For example, among many subjects, the most dramatic alterations seem to occur in the skeletal and muscular systems. In Group A subjects, it appears that the initial HVV infection triggered the production of parathyroid hormone-related protein, which typically results in the gracile elongation of bones. LPV, on the other hand, typically activates dormant osteoclasts and chondrocytes in the epiphyseal plates, leading to rapid bone resorption and reformation. In the co-infected subjects, it appears that this process was moderated by the pre-existing HVV changes. The result was a unique bone structure, longer than typical lichens due to the initial HVV elongation, but denser and more robust due to LPV's influence on bone mineralization. Group B subjects experienced a different progression. The initial LPV infection triggered rapid bone growth and restructuring as expected, but when HVV was introduced, it appeared to interfere with LPV's osteogenic processes, particularly in the facial bones. This interference resulted in a less pronounced muscle shape compared to pure lichens, as HVV's influence seemed to partially counteract the extreme elongation of the maxilla and mandible. Furthermore, muscular hypertrophy in both groups exceeded what is typically observed in either pure vampires or lichens. HVV normally increases the proportion of fast-twitch muscle fibers and enhances mitochondrial efficiency, while LPV triggers widespread protein synthesis and the proliferation of muscle cells. In co-infected subjects, these processes appear to compound each other. The enhanced mitochondrial function allows for greater energy production, fueling the excessive protein synthesis and cell proliferation. Additionally, the increased fast-twitch fiber ratio is maintained even as overall muscle mass grows, resulting in subjects with both extreme speed and strength. But perhaps the most intriguing changes occurred in the circulatory system. In Group A subjects, the initial HVV infection began the process of cardiac atrophy and the transition to skeletal muscle-driven circulation. However, the introduction of LPV, which typically causes cardiac hypertrophy due to the increased metabolic demands of the lichen form, disrupted this process. In co-infected subjects, this led to a partial reversal of the HVV-induced cardiac atrophy. In some cases, the result was a hybrid circulatory system where both cardiac and skeletal muscles contributed to blood flow. But while this dual-driven circulation appears to move blood throughout the body quite well, it also led the subjects to bouts of fluctuating, uncontrollable spasms, far worse than the tremors experienced by vampires. Olfactory capabilities were universally enhanced beyond levels seen in pure infections of either virus, which appears to be due to a combination of HVV's enhancement of olfactory bulb sensitivity and LPV's enlargements of the olfactory cortex. However, this hypersensitivity often caused distress due to sensory overload. At this point, we've discussed at length the changes observable at the macro level, but of course, these outcomes are the result of what occurs at the cellular level. HVV, as an RNA virus, typically infects cells through endocytosis and alters cellular function to produce viral clones without immediate cell destruction. LPV, a DNA virus, integrates its genome directly into the host cell's DNA. In co-infected cells, these processes appear to interfere with each other. HVV's alterations to cellular metabolism seem to make it more difficult for LPV to successfully integrate its genome. Conversely, in cells where LPV has already integrated, HVV's replication is often impaired. This cellular conflict results in a patchwork of differently affected tissues throughout the body, explaining the often asymmetrical and variable nature of the transformations observed. It also likely contributes to the high mortality rate, as the competing viral processes place enormous stress on cellular function. The resulting creatures possess strengths and abilities that exceed those of either vampires or lycans alone, but also suffer from instabilities and weaknesses unique to their condition. The implications of such creatures existing in the world are, to put it mildly, deeply troubling. The next phase of the study was a natural progression, and some months later my assistant and I began the arduous task of procuring yet another sample. We selected 30 subjects, again focusing on individuals between 25 and 40 years of age, all in good physical condition. This time, all subjects were to be infected with both viruses simultaneously. To achieve this, I prepared a solution containing both HVV and LPV in equal concentrations. The solution was administered via injection to ensure precise dosage and timing. Within 2 hours of administration, all subjects began exhibiting signs of severe physiological stress. The speed of this response was unprecedented, leading me to believe that a synergistic effect between the two viruses accelerated the initial stages of infection. During the first 12 hours, subjects experienced hyperpyrexia, likely the result of both viruses triggering separate immune responses and compounding the body's natural reaction. Cardiovascular instability was rampant, with subjects alternating between tachycardia exceeding 150 beats per minute and bradycardia dipping below 40 beats per minute. Furthermore, subjects reported intense, widespread pain deep in their tissues, which I suspect was due to the simultaneous onset of HVV's cellular alterations and LPV's aggressive tissue remodeling. Neurological symptoms were also prevalent, and seizures affected a majority of subjects within the first 12 hours. The metabolic impact was equally severe, manifesting in rapid weight loss. Unfortunately, the mortality rate during this initial phase was staggering. Within the first 24 hours, 8 of the 30 subjects expired, their bodies seemingly overwhelmed by the competing viral processes. Autopsies revealed severe cellular damage across multiple organ systems, with evidence of both HVV's more subtle alterations and LPV's more aggressive restructuring occurring simultaneously in many tissues. As the surviving subjects moved past the initial 24-hour period, greater variations in their transformations became apparent. Unlike in the study of sequential infection, here there was no clear pattern to the expression of vampiric or lycanthropic traits. Many subjects exhibited stark asymmetrical changes between the left and right sides of their bodies. One subject, for example, developed the elongated fingers characteristic of HVV infection on their left hand, while their right hand began to show signs of the thickening and claw-like nail growth associated with LPV. This physical dichotomy extended to other parts of the body, creating bizarrely mismatched features. Sensory abilities in the subjects fluctuated rapidly, and many were often severely disoriented. Some began to exhibit rapid hair growth, only to have it fall out hours later. Perhaps most disturbingly, subjects' cognitive functions seemed to oscillate between the heightened intellect associated with HVV and the more primal state induced by LPV. This led to periods of lucidity interspersed with bouts of feral behavior, often shifting rapidly and unpredictably. During their lucid moments, subjects often expressed horror and confusion at their condition, only to lose coherence mid-sentence as their minds were overwhelmed by more basal impulses. As the study progressed beyond the initial chaotic phase, the surviving subjects began to exhibit more stable, though no less disturbing, transformations. Of the 22 subjects who survived the first week, 18 remained alive after 1 month, each a unique amalgamation of vampiric and lycanthropic traits. The stabilization process itself was a harrowing experience for the subjects. Many experienced periods of intense pain as their bodies settled into their new forms. Bones creaked and shifted, muscles tore and regrew, and skin stretched and reshaped itself. Once again, the subjects' agonized cries reverberated through the facility, each echo cutting to my soul. But eventually, the transformations did stabilize, and interestingly, as they did so, 3 distinct categories of hybrid beings emerged. I have termed them alpha, beta, and gamma types. The alpha types, comprising about 30% of the survivors, exhibited a predominance of vampiric traits. These subjects retained a more human-like appearance, with pale skin and elongated limbs characteristic of HVV infection. However, they also displayed some lycanthropic features, such as increased muscle mass and enhanced olfactory capabilities. Their eyes were perhaps their most striking feature, combining the hyperdilated pupils of the vampire with the gold coloration typical of lycans. Alpha types retained much of the heightened intelligence associated with vampires, but also exhibited periods of primal aggression more typical of lycans. These episodes were less frequent and severe than in other hybrid types, but when they occurred, the contrast with the subject's usually calculated demeanor was deeply unsettling. Beta types, making up approximately 40% of the survivors, represented a more even blend of vampiric and lycanthropic traits. These subjects underwent more extensive physical changes, developing a form that could be best described as a more sleek, predatory version of a lycan. They exhibited limited hypertrichosis, somewhat elongated jaws with pronounced canines, and partially digitigrade legs. However, they retained the graceful movement and pale skin tone associated with vampires. Cognitively, beta types were the most unstable. They oscillated between states of vampiric cunning and lycanthropic ferocity, sometimes shifting between the two in a matter of moments. This cognitive instability made them particularly dangerous and unpredictable. The gamma types, the remaining 30% of the survivors, leaned more heavily towards lycanthropic traits. These subjects underwent the most extreme physical transformations, developing a form more bestial than human, but retaining unsettling remnants of their humanity, such as less hair growth and less pronounced facial remodeling. They also appeared to exhibit some vampiric characteristics, such as increased speed and agility beyond that of typical lycans. Surprisingly, gamma types showed the most stable cognitive function, albeit at a level below human baseline. They seemed to settle into a state of primal cunning, combining the predatory instincts of lycans with a shadow of vampiric intelligence. One of the most intriguing findings across all types was related to the PCDH-Y protein. In pure HVV infections, the inability to synthesize this protein necessitates the consumption of human tissues. However, in our hybrid subjects, we observed varying levels of PCDH-Y synthesis capability. Alpha types reflected HVV victims' inability to synthesize the protein at all, while gamma types retained the ability at near normal levels. As a result, alpha types required the most human tissues, such as blood, in their diet, while gamma types, much like traditional lycans, were not picky at all, though they certainly required many thousands of daily calories to meet their metabolic demands. Another interesting aspect of our hybrid subjects was their varied responses to the crucifix glitch, a phenomenon observed in vampires when intersecting right angles occupy more than 30 degrees of visual arc. Alpha types exhibited the strongest reaction to these right angles, experiencing disorientation and seizure-like episodes, though less severe than in pure HVV infections. Beta types exhibited a more muted response, displaying discomfort and agitation, but rarely progressing to full seizures. Gamma types showed the least reaction, with most exhibiting only momentary disorientation, if anything at all. I suspect that the varying degrees of susceptibility to the crucifix glitch among hybrid types is simply due to the level of predominance of vampiric traits in those types, particularly in regards to the visual cortex. I attribute the near immunity of gamma types to the glitch to the more extensive remodeling of brain structures by LPV, which likely bypasses the specific neural circuits responsible for the crucifix glitch in HVV infections. By the time these studies concluded, both my assistant and I were utterly exhausted, both physically and mentally. There is no solace from the horrors we've experienced, nor that of our subjects. There is no respite from the screams that still echo in my mind. There is only the small hope that this work will finally shed light on the threats that face all of humanity should these pathogens be disregarded as mere fiction. Indeed, the level of adaptability displayed in many of these subjects exceeds that of pure vampires or lycans, which would undoubtedly make predicting and countering their behaviors significantly more challenging. Worse, many hybrids exhibited resistance to traditional weaknesses of both vampires and lycans, which make them exceptionally difficult to control or eliminate. Indeed, we may see wild populations of intelligent lycans and hyper-aggressive vampires, either of which could accelerate the inevitable crisis these viruses would bring upon the greater population. As for myself, I no longer possess the resolve to continue studies of this magnitude. I've done what I can, and now the result is up to you, dear listener. Even the light of scientific discovery I so revere is growing dim, and the world is getting darker for it. I implore you to act now, before that flame is extinguished forever.